Hello, and welcome to section 4.4 of Clark, Choi, and Douglas's textbook on biology, second edition. This textbook seems to be open source, so I'm trusting that they don't mind me using some of their diagrams as I promote their materials. Well, it is section 4.4, the endomembrane system and proteins. So we're inside the cell. And I find this fascinating. So here's their diagram. It's a lovely diagram. Thank you, Clark, Choi, and Douglas. I could have drawn it, but it wouldn't have looked anything like this. It wouldn't have been nearly as good. So this is inside the cell, right? It's only a small portion of it. And inside the cell, there is the nucleus. Think of this, I think of this as a kind of assembly line. Now, the nucleus hands the mRNA to the ribosomes. These are ribosomes, little raisins in the pudding there, little dots there, like a little raisin in my oatmeal. Sophie doesn't. I don't know why, but anyway. So this is the endoplasmic reticulum, which basically means the plasma network. So endo means in, it's in the plasma, where, we've got this cytoplasm of this brownish, is it? Cytoplasm is the liquid, the gooey stuff. And then this is so it's an in the plasma network or net or system here. It's just passing it along. Basically, proteins are going, and then they either leave the cell or they become part of the membrane. And so this is kind of like a train. We're just carrying it. We're passing it along, the proteins, and they're getting modified. It's kind of like an assembly line. So add this here. Okay, we're putting this on there. So we started with this protein. Now we're putting on some carbohydrates. Now, and we build this membrane. And by the time it gets added to the outside of whatever it is. So think of this system, this endoplasmic system, membrane system. Think of it as a kind of assembly line that's taking proteins and then modifying them and kind of transporting them out to the cell, either to the cell membrane or spitting it out into the world. So the nucleus is connected to the rough endoplasmic reticulum. The rough endoplasmic reticulum is spotted with ribosomes. The ribosomes send proteins to these vesicles, which are like taxis. The vesicles takes taxi to the Golgi apparatus. That they attach stuff to the proteins in their cisternae, and then more vesicles taxi to the cell membrane. Now hear the word of the Lord. Okay, nobody knows that song who's listening to this video. Anyway, so the nucleus sends the mRNA, I think it is, that basically tells the ribosomes, here's the order. So the ribosomes are the cooks, flipping the burgers, flipping the proteins. So all these ribosomes in the rough endoplasmic reticulum, by the way, there should be, I think, smooth endoplasmic reticulum that doesn't have ribosomes on it. This diagram doesn't show the smooth RNA, but apparently, so the endoplasmic reticulum, which is these folds, it's just basically a transport. It's like the things just going through the line or being handed off to the next person, to the next person. These are just basically transport lines. Think of it as a maze. We're sending proteins down the maze or down to down the hole, and it's just being handed off from one person to the next until it finally gets out to the membrane. So the nucleus sends the order to the ribosomes. Here's we want a hamburger with medium with mustard and mayonnaise. And so the ribosomes make the initial protein. They make the burger. Now we're going to put stuff on the burger. So like here, this is a carbohydrate. being attached to a protein. And so, I don't know, it's green. Think of it as the lettuce. We're going to add lettuce to the protein. And then this is a vesicle. A vesicle is like a taxi. Taxi, I need to get to the Golgi apparatus. And so the protein, now modified, gets in the taxi. The RER, the rough endoplasmic reticulum, has all these little fry cooks cooking the burgers. And then they send the proteins to the vesicles, the taxis. The taxis take it to the Golgi apparatus. Now these folds, so these are folds. These are called lumen, I think, the folds in the ER. Emergency room. No. Anyway, feel free to try to do something with that. So the Golgi apparatus modifies it. Actually, the Golgi apparatus tries to sort. Oh, this goes over here. Okay, put that over there. So the Golgi apparatus kind of sorts the stuff that's been created in these cisternae, the sisters of the Golgi. For whatever reason, this side of the Golgi is called the cis face, and then the part facing the membrane is called the trans face. So I don't know, cisgender means like I'm cisgender because I'm biologically male and I identify as a male. So I'm cisgender. It seems to mean same. So I guess the cis face is facing the endoplasmic reticulum, but it ends up trans by the time it gets to the other side. This is, okay, bad joke. Anyway, so this side is the trans side facing out. And so again, the Golgi apparatus sorts, so the vesicles taxi to the Golgi, and then stuff is attached. They get modified. They get pickles and mustard and mayonnaise in here, and they get sorted. And then a vesicle takes them to the cell membrane, and voila, either it becomes part of the cell membrane or it gets flushed out into the world of outside the cell. So this is a relay. Here, drop. They hand the baton, and then the next takes up the baton, or think of it as a sandwich operation. The order is made here. Ribosomes make the protein. Lettuce gets added. It gets sorted. These are the people who have the medium burgers. These people have the rare burgers. It's all sorted, and then it gets on another taxi, another Golgi vesicle, and taken out. Okay, so the process again. Ribosomes make the proteins in the rough endoplasmic reticulum. They get modified within the rough endoplasmic reticulum in those folds that are called the lumen. And then there's a smooth endoplasmic reticulum that doesn't have ribosomes, but it continues to modify chemically. These are chemical additions, right? Functional groups, macromolecules. It all gets very complex. And then the Golgi apparatus sorts them. It starts on the cis side and it leaves on the trans side. Okay, the last thing in this section are lysosomes. Lysosomes are the janitors of the cell. They clean it up. So they destroy pathogens. Pathogen are the things that are they're going to cause the cell to get sick. Things that aren't healthy for the cell. So lysosomes basically capture them and put them in jail and then destroy them. So as an example, in white blood cells, the white blood cells in our blood are the blood cells that kill diseases. And there's a certain group of white blood cells that are called macrophages. Now, macro means big, and phages means to eat. So the esophagus is comes from a Greek word, actually. The root is phag, and it means to eat in Greek. And so these are the big eaters, the macrophages. They eat stuff, and in particular, they eat bad stuff, pathogens. They bag it, they surround it, they put it in a bag, and then they destroy it in the bag. That's what they do. So again, thank you to the book for this lovely diagram. So here's a picture of a macrophage. And the process of destroying these pathogens is called phagocytosis. Again, phag means to eat in Greek. So it's the eating of the stuff. So the food particle goes in, a vacuole kind of bags it, and then the enzymes break it all down, and then they poop it out of the cell. And so that's kind of how lysosomes. So these would be, I think these represent the lysosomes in the cell that surround. So of course, the food particle here isn't a pathogen, but this is the process of, so it can be digesting good stuff also. But this is basically the way the macrophage takes care of both pathogens and food that it's digesting. Well, I gave it a shot. This has been a video on the endomembrane system of the cell, transporting and modifying proteins, starting in the ribosomes where they're created and transporting them to the cell membrane and beyond.